Okay, it is 2 o'clock and it is time to start the webinar. My name is Hanne Ørnsted Hansen and I'm a coordinator at Danish Diabetes Academy. We are delighted that you have joined the webinar with Professor Gernot Desoye from Medical University of Graz. And of course, thank you to Professor Desoye for accepting to present at this webinar. Just a few practicalities before we start the presentation. The presentation will be approximately 20 minutes. That leaves 10 minutes for questions and discussion. And I know Professor Desoye is looking forward to that part of the webinar, so do not hesitate to pose your questions. If you have a question, please use the chat function. If you mouse over your screen and click at the chat icon, the chat panel will open to the right. And I can see some of you have already found the function. You can either write your question or you can write, I have a question or something similar, if you prefer to ask your question verbally. And then I will unmute you. And it will be very helpful if you write your questions to all participants. That was all for me, and now Professor Desoye will start his presentation. I hope you will all enjoy the webinar.
So thank you very much, Hanne, for organizing and arranging this webinar, and thanks to all participants for joining and being interested in something that has been at the core of my own interest over the past several decades, I have to say now, and that is addressing the question how the placenta, particularly the human placenta, affects the fetal growth, and in particular fetal overgrowth that we all see in pregnancies complicated by diabetes and or obesity. And in this webinar, I actually want to raise your awareness that the first trimester or the early pregnancy period is really important in playing a role in determining placental growth, but ultimately then fetal growth. We all know that type 1 diabetes and pregnancies complicated by type 1 diabetes have been characterized a long time by a smaller neonate, but there is accumulating evidence now that over the past decades, the birth weight has increased. As you can see here, there's data from Scotland that show the birth weight Z-score over almost 20 years period. And also the proportion of large for gestational age offspring in Swedish studies has increased from 20% in the 80s to now almost 50% in the 2000s. Now, it is important to keep in mind that the fetal growth is biphasic in type 1 diabetes. That means offspring with elevated growth, higher birth weight, tend to have a lower weight or lower growth, and this is biparietal diameter that was measured here in this ultrasound study in the 89s. Early on in pregnancy. So that would suggest that there is indeed reduced growth early in pregnancy, followed by a catch-up growth later in pregnancy, resulting ultimately in an increased birth weight in the neonates born to pregnancies complicated by type 1 diabetes. Now, as a placentologist, one always tends to think the placenta is key to many of the outcomes of a pregnancy, and the question that I've been interested in is whether now the placental growth is also biphasic, and if it's reduced in pregnancies complicated by type 1 diabetes. And there is evidence in the literature to suggest that this is indeed the case. In diabetic rats that were rendered diabetic prior to conception, and the weight of the placentas was followed then throughout pregnancy, then you can see the placental weight is reduced early in pregnancy, followed by rapid catch-up growth later in pregnancy, And ultimately, at the end of pregnancy, placental weight is higher in the diabetic animals as compared to the control animals. And this does not only apply to placental weight, which may have been due to an increase in the water content, but as you can see here on the right panel, there is also the same pattern or the same dynamics can be seen for the total protein content. So the question is, does this also occur in the human? We know that the first trimester is the period of most rapid placental growth in human. This is on the left panel, you see the dynamic changes of placental weight over the course of pregnancy, and there is almost a linear increase. However, I calculated also the relative weight change per week and plotted it here, as you can see on the right panel. And it is very obvious that it's really the period around week 10 to 12 to 14 where placental growth is most rapidly occurring. And we know that rapidly growing tissues are actually very sensitive to environmental perturbation. So that raises the question whether the metabolic changes or actually all the environmental changes associated with type 1 diabetes may have an effect on placental growth early on, and in particular may have, as a result, a reduced placental growth. In fact, there is some indirect evidence that this is true. The concentration of placentally derived hormones such as placental lactogen and pregnancy-associated plasma protein A are lower in the maternal circulation, and these proteins are synthesized by the syncytial trophoblast, which is one of the key endocrine tissues of the placenta, as opposed to hormones that are produced by the endometrium, and the concentration of which is more or less unaltered in the serum of mothers in type 1 diabetes as compared to non-diabetic pregnancies. So there is this indirect evidence that placental growth may be reduced in first trimester diabetic pregnancies. And we have some unpublished data that we could generate in a cohort of first trimester placentas from type 1 diabetic pregnancies and controlled pregnancies. And what we did was just to measure, in this graph, to measure a simple proliferation parameter, Ki67, by Western blotting. And as you can see here, there is a significant reduction in these Ki67 signal in the Western blots of placentas obtained from type 1 diabetic pregnancies as compared to controls in the first trimester. And that is something that really is confirming this idea that there is a reduced placental growth early on. And I have to emphasize that the data were obtained here in week 7 to 9. And I will come back to the gestational age relevance of gestational age a bit later. So the question is now, if there is a reduced placental growth in type 1 diabetes in the first trimester, what may account for this? And of course, the first idea that we have come up with was that hyperglycemia may be one of the drivers of reducing placental growth. And in order to address this, we isolated trophoblasts from the first trimester placenta and exposed them to 25 millimolar glucose versus 5.5 millimolar glucose, which served as the controls. And we exposed the cells to the glucose for 24 hours and also for 48 hours. And as you can see here, there is a difference and there's a reduction in the number of viable trophoblasts when the trophoblast was exposed to hyperglycemic conditions. And that was independent of the gestational age. We've separated the data here to see whether there was a differential response to the hyperglycemia here in week 7-8 as opposed to week 11-12, but the results were more or less the same. So that again would suggest that there is a reduction in trophoblast growth, which is the main driver of placental growth early in pregnancy, and that this might be induced by hyperglycemia. So the question is now, would a hyperglycemia early in pregnancy also, in type 1 diabetic pregnancies, also relate to a higher birth weight? And in a study that was recently published, Rosa Corcoran's group demonstrated that the self-monitored blood glucose levels in the first trimester of pregnancy did not associate with birth weight. But when they did the same analysis for the second and third trimester, it turned out that in the second trimester, the self-monitored blood glucose levels in type 1 diabetic women associated with birth weight. And that would somehow suggest that there may be an effect of hyperglycemia on early placental growth, resulting in reduced growth. And that once the placental constraint on fetal growth is released, that then the hyperglycemia may stimulate fetal growth by whatever mechanisms in the second trimester and later on. But glucose is not the only metabolite that may have an effect on placental growth, the second one that we have been interested in was insulin, and asking the question whether hyperinsulinemia may have an effect. And this was somehow sparked by observations that Pet Catalana's group published some 5 years ago. They looked into the potential association of insulin concentration with placental weight at the end of pregnancy, and analyzed the results for 3 different periods. They measured the insulin response to an intravenous glucose tolerance test pre-pregnancy and did not see any association with placental weight at the end of pregnancy. The same result was found when they tried to associate the insulin response of the IV GTT to placental weight late in pregnancy. But interestingly, there was a positive and significant correlation of this insulin response in early pregnancy, defined here as week 12 to 14 weeks, with placental weight at the end of pregnancy. So there seems to be at least some association of insulin levels, if you so will, early in pregnancy with placental weight at the end of pregnancy. So insulin may drive placental growth that will then track through pregnancy until the very end. Insulin, interestingly, the daily insulin dose given to type 1 diabetic women early in pregnancy here in the first trimester, that associated with birth weight. So insulin does seem to play a role in driving not only placental weight or placental growth, but also fetal growth. The effect of insulin on the placenta is can be explained by the presence of insulin receptors on the trophoblast surface, in particular on the microvillus membrane of the syncytial trophoblast. As you can see here, this is immunohistochemistry standing for insulin receptors. This is the microvillus membrane. This would be intervillus space where maternal blood is circulating with the insulin, and insulin will then be able to interact with the insulin receptors present on this microvillus membrane of the syncytial trophoblast in the first trimester. The effect of insulin, however, on placental growth, if any, cannot be a direct effect, but has to be an indirect effect. And the reason is the following. The syncytial trophoblast is, which is here the tissue layer in gray. This would be maternal blood with the insulin. Here are the insulin receptors. The syncytial trophoblast is not the growing tissue in the placenta or proliferating tissue in the placenta, but it's the layer of the subjacent cytotrophoblasts. And in the first trimester, there is a more or less continuous layer of the cytotrophoblasts, which serve as the sort of stem cell for the syncytial trophoblast. So the cytotrophoblasts, they proliferate, And once they have exited the cell cycle, they can then fuse with a syncytial trophoblast and thereby enlarge the syncytial trophoblast. For a particular reason, we got interested in an enzyme that is called matrix metalloproteinase 14, which is a membrane-bound MMP. Which we found on the surface of the cytotrophoblast. And in vitro studies, we demonstrated that insulin, excuse me, insulin through the activation of the protein kinase B pathway can activate this enzyme MMP14. And we could also demonstrate that MMP14, once activated, facilitates the fusion of the cytotrophoblast with the syncytial trophoblast. So this could then represent an indirect mechanism by which maternal insulin, through activating MMP14 on the surface of the cytotrophoblast, leads to a dissolution of the membrane of the cytotrophoblast and fusion of the content of the cytotrophoblast with a syncytial trophoblast, and ultimately resulting in an enlarged placental surface. And this MMP14 is present in higher amounts in the placenta in the first trimester of type 1 diabetic pregnancies. So that would argue for a positive effect, a stimulatory effect of insulin through MMP14 on fusion of the cytotrophoblast to syncytial trophoblast, thereby enlarging the trophoblast surface. And this is not only supported by the in vitro data that we have extensively carried out, but we also try to see whether there's some sort of in vivo evidence for this. We just correlated the total MMP14 levels in the first trimester trophoblasts of the, in the first trimester placental tissue of type 1 diabetic pregnancy with daily insulin dose. And as you can see here, there is indeed a correlation. So that would somehow suggest that insulin may indeed play a role in regulating placental growth early on in pregnancy. So that leads us to the concept that we already hypothesized very early on, 6 years ago, that a disturbance of the insulin glucose axis in the first trimester may affect placental growth. And this by whatever mechanisms, and there is a big question mark here, of course, can then lead to a change in the glucose insulin axis in the fetus, ultimately resulting in an increase in the proportion of LGA or the number or the proportion of neonates born with elevated birth weight. So in this concept, we have put emphasis on insulin, but of course there is a strong interaction between insulin and glucose. Now, this is of course much too simplistic a view, and there is certainly additional factors in the first trimester operating to influence the outcome of the pregnancy. One of the many unknowns that we have here is how does placentation occur? Is placentation affected by the diabetic environment? That might then result in a change in the remodeling of the spiral arteries through which maternal blood flows to nourish the fetal-placental unit. This remodeling of the spiral arteries is important because it allows, after it has finished occurring, it allows the flow of fully oxygenated blood from the mother into the intervillous space, thereby increasing the oxygen tension in the intervillous space. And we know that oxygen is a powerful regulator of all kinds of biological and metabolic and biochemical events. And because of this increase in the oxygen tension in the intervillous space, between the early period of the first trimester around week 6-8 and completed by week 10 to 12 in the first trimester. It is important to really also address the question of the influence of gestational age and to always consider if there is an effect of gestational age. This definitely complicates matters, particularly when one wants to do in vitro studies, but also in vivo studies, in clinical studies. One will have to distinguish between various periods, even within the narrow period of the first trimester. And in fact, in this study that I have alluded to by Rosa Corcoy's group, they found that it was the period of week 1 to week 7 in the first trimester in the type 1 diabetic pregnancies, where the insulin levels associated much stronger with birth weight than the insulin or the daily insulin dose of the women in the period of week 8 to 12 or 8 to 13 in the first trimester. So that again supports the concept that also gestational age, even within the first trimester, is important to consider when one carries out studies. And then, of course, there's the question about potential effects of additional metabolites. And my current thinking is that we should pay attention to amino acids, which can serve as potent insulin secretagogues for the fetus. And ultimately, and finally, there is also the question about the effect of fetal sex. We know that the outcome of pregnancies is different between male and female fetuses. And also the placental response to all kinds of environmental perturbations is different depending on the sex of the fetus. And it has been shown by Maria Pisarska's group from Los Angeles 2 years ago in the biology of sex differences that the placental transcriptome differs between males and females already very early in pregnancy, and they measured it at, between week 11 and 13. So sex differences are also very important to take into account when we want to understand the role of the early pregnancy period on fetal growth and pregnancy outcome. So with this, I want to thank you, and for your attention. I want to encourage everyone to come up with questions, which is actually always the fun part of giving talks and presentations. Thank you very much.
Thank you. And yes, we are ready for questions. And please use the chat function to ask your questions. You find the chat function if you just mouse over your screen or click at your screen and click at the chat icon.
And please don't be shy. I always like challenges, but I also like input.
So comments or questions. You can either write your question or you can write something like, I have a question. Then I will unmute you and you can ask your question verbally.
Also, if you want me to clarify things or aspects or have other questions, yes, not directly related to what I just said, just come up.
I have one person saying I have a question. I will unmute Alison Petition. You are now unmuted.
Hello, am I unmuted?
Yes, hello, we can hear you.
Hello, I'm Alison Petition from Lausanne Nestlé Research. And I had a question because you had shown that the placenta has the insulin receptors. And I was wondering if in the case of type 2 diabetes, where in addition to an increase in insulinemia and glucose, we also have an increase in inflammation, or in other LDLs that are going around in the blood as well. Could we imagine that this increase in inflammation in the placenta, if that happens, could this cause an insulin resistance in the placenta, much like it does in the liver?
Yeah, thank you for the question. That's interesting. We have been thinking about how to measure this. We are currently doing it but the problem is always it will require in vitro work. So you will have to isolate trophoblasts, and once you isolate trophoblasts, and even if you keep them at low oxygen tension, you will always induce some sort of stress, and we know that stress affects insulin signaling. So we can measure something, and but I'm not sure if that would reflect true biology. What I can tell you is that in obese or in pregnancies with obese women, it was Silvio Gell's group in Cleveland showing that the response of insulin, the response of the trophoblast to insulin was much lower in trophoblasts isolated from obese versus lean women. And that would somehow suggest that there is a bit of an insulin resistance, but it definitely requires further work and clear demonstration.
Yes.
So there was a question about any relation to first trimester miscarriage. Yes, I would think there is a relationship between type 1 diabetes and first trimester miscarriage. The mechanisms, I think, are still unclear, but I would, let's say, hypothesize or speculate that it's related to effects on implantation and trophoblast invasion. The next question was about, you mentioned amino acids. What is your take on microRNAs and the role in regulating the insulin axis? Yes, I bet there is a role for the microRNAs. I'm not sure if anyone has ever looked into the first trimester. We know that the microRNAs is all the other, or many of the other RNAs are expressed in a sex-dependent manner. So one would have to look at sex differences. One would have to take into account the gestational age period, etc. So it's if someone wants to work on type 1 diabetes or in the first trimester or on placental development in obesity in the first trimester, we do have tissue in our lab, and we are currently focusing all our efforts to analyze this, but we're a bit short of hands. There was another question, a clarification question. You mentioned that amino acids will play. Did you mean insulin-trophic amino acids, specific diets with certain times of pregnancy? I meant actually arginine, leucine, which are insulin secretagogues and which have been demonstrated to be the first insulin secretagogues for the human fetal pancreas. So they stimulate insulin release earlier than glucose. So this is why I thought that amino acids may be important to consider and measure the uptake of amino acids, particularly these amino acids, into the first trimester trophoblast or placenta. How do you think your findings will be if it was gestational diabetes or type 2 diabetes? Well, I think there will be some similarity. There is evidence now that also fetal growth in pregnancies that later develop gestational diabetes is reduced earlier in the earlier pregnancy period, followed by this catch-up growth. So there may be some similarity. But of course, as regards placental growth, we don't know, because it's one would have to do longitudinal ultrasound studies on placental volume, etc., in pregnancies, and that will later develop, where the mothers will later develop gestational diabetes. There was, I'm sorry, how do you think if I was to, which parameter do you believe is the most determining factor on first trimester placental growth? Hyperglycemia, placentation, gestational age, metabolites, etc. Thank you for this question. I wished I knew. I think it's not one parameter, but it's certainly a network of parameters, and the power of the one will certainly depend on the whole network. Therefore, I think this is a question that we are far from being able to even speculate about. And which parameter do you think is the most important for later fetal growth? Well, for me, it's certainly the insulin in fetal circulation, because that is of all what we know is the insulin driving fetal fat accretion, particularly later in pregnancy. Yes, of course, there was someone wanted to have the PowerPoint. I don't know through which mechanism we can share the PowerPoint, but of course, I can do that. Do you think
I will send the PowerPoint to all the participants. That's
all
no
right.
problem.
So I can send the PowerPoint to Hanne, and you will share it. Good, thank you. So do you think that insulin also plays a role even in healthy pregnancies? For example, is placental weight higher in women with higher insulin levels compared? Yes, there is evidence that fetal insulin or insulin levels in the cord blood associate with placental weight at the end of pregnancy. There is, however, situations in which the fetus does not produce any insulin. That is in the case of pancreasogenesis, and then there is a reduction in placental weight, but it's only about 25 or 30%, if I remember correctly. So insulin does play a role in also, or I would suspect that insulin plays a role, and these associations may support this idea late in pregnancy, but it's certainly not the only factor. Can you talk about whether there could be a connection between the observed reduced growth of placenta and lower HbA levels in early type 1 diabetes and the increase in insulin sensitivity often seen at that same time? Yeah, placental lactogen is of course stimulating the pancreas, but there is not only placental lactogen secreted early on in pregnancy as one of the hormones that facilitate metabolic adaptation of the mother, but there's others as well, and HCG and many others. So I think we should not only focus on one or two of these regulators for insulin sensitivity or pancreas function early in pregnancy, but also look at a broad spectrum of these regulators. Could you speculate on how we could implement this knowledge, especially regarding the basic growth in a clinical setting in pregnant women with pre-existing diabetes, for example, regarding induction of labor, pregnancy planning? Sorry, but this is beyond my absolute expertise. I'm always daring to speculate, but this is too far out of my comfort zone, so I can't help you here. Sorry.
Earlier on, there was a question from Line Hjort. I will just copy it and then.
Oh, I'm sorry, did I miss it?
Oh, yeah, no, there was a lot of questions suddenly, but now the question should be at the bottom.
Dear Gernot, do you think GDM have similar mechanisms on the trophoblast and placental weight as in type 1? Was that the question?
Yes, exactly.
Yes, I do think that there is similar mechanisms in place, although gestational diabetes or the metabolic changes in pregnancies in the first trimester that will later on lead to gestational diabetes are certainly much more subtle. But I do think there is evidence that hyperglycemia in the first trimester, maternal hyperglycemia associates with the proportion of neonates born LGA, and also with a proportion of women developing GDM. So it always comes down to the glucose insulin axis and to insulin sensitivity, particularly in the first trimester. And we do have some supporting evidence that we are about to hopefully publish pretty soon, that it's actually the insulin sensitivity in the mother that is playing a key role here.
Okay, I will just post one more question. That was from a participant who posted her question to me, but it should be at the bottom.
Yeah, can you comment on the implications in type 2 diabetes with hyperinsulinemia? I'm sorry, I'm totally out of knowledge. I mean, type 2 and GDM are pretty similar, so I would just suspect that the metabolic changes in early pregnancy in type 2 diabetes are more pronounced than in pregnancies where the mothers are gonna develop GDM later in pregnancy. And the effects that the environment will have early on, hence will probably be also stronger than in pregnancies where you have GDM later on. But I'm not aware of any literature, and there is actually very, very few people studying first trimester placenta because of difficulties in getting hold of the tissue. And I'm not aware of anyone looking into type 2 effects on early pregnancy.
Thank you. Do we have a last question? Or doesn't seem so? Then I want to thank you all for joining the webinar, and thank you for the presentation. And
Thank you.
as agreed, I will send you the PowerPoints to you. Bye-bye.
Good. Thank you. Bye-bye.
Bye-bye.
Okay, and we'll send you the PowerPoint now.
That would be great. Then I can send it to the participants.
Yes, good. Thank you very much.
Yes, thank you.
Thank you for your support. Bye-bye.